Okay. Hi, I'm Josh. I'm in the BME department. I wanted to plug some of the work in the lab, but I guess what you should picture is maybe talking to, you're going to see a lot of different types of analyses and work, but I guess you'll have to trust me that it all does relate. It's all connected, dude. So just hang on to the end and to see the connection maybe. But so the thread will be how we can use the transcription RNA coming from the RNA sequencing to learn about tumors, and the current group, and there's some past members in here whose work I'll talk about to plug some projects going forward that I'm looking for new students to work on. So anyway, this is the lab, and you'll hear me talk about a work from Chris Wong in the group, who's still with us, Verena, Bianca. Ding is like a co-PI, he's a postdoc in the lab who helps me coordinate and advise on these projects. But also mentioned, so Vlado just recently graduated and David Hahn recently graduated, and their work is featured prominently in some of this stuff. So as a launching point, I want to mention sort of a basic but still important analysis in cancer genomics. So we're looking for mutations and variations, obviously, genomic changes and lesions that underlie patient tumors. Just finding these things and teasing them out from noise is a tough problem, and so on. People have called it like finding a needle in a haystack. It's more like a needle in a needle stack in a sense, just to find these things. And then 10 years ago now, as part of these consortia, national and international, the UCSC played a prominent role. I just wanted to point this out just to rewind and get a perspective that it used to be we would run everybody's mutation callers. Those are each of these circles. And the overlap wasn't so great. It'd be like half of the mutations called across all these tumor types overlapped. And everybody'd be like, well, that's even for single nucleotide variations. It was worse for structural variations, right? So the middle intersection is the agreement, right? 500 in agreement out of thousands called from these methods, right? Me, Hausler, a lot of folks worked over the years to develop unified callers and ran competitions, and I won't go into all that. I've given talks in the past on that, these dream competitions, sorry. But eventually, we have lots of different methods now, and we can integrate them with machine learners to get better predictions. And I guess as of a couple years ago now, and this was actually used in a international version of this project that was published this last February. The punchline is that we now have callers. The first thing is how many mutations are called, and you can see that the majority now are in agreement across these 5 different methods developed from different groups and so on. That's what this plot is supposed to show you. And this is run on over about 30 different tumor types. Anyway, so that's cool. And that's sort of led to these the variations and so on have led to these projects that we've participated in that look across tumor types, lots of different tumors, integrating different pieces of information to interpret when a mutation is a driver versus a passenger. So when it actually looks like it leads to an effect versus it's just there and got altered because of the mutational processes have increased in a tumor and so on. And this is what we've termed the pan-cancer projects that our lab and other folks here at the university have worked heavily on, like Jing Zhu's group and her Xena browser can show you all that information. And David's group worked on a lot of the genomic interpretation, and my group worked on some of the pathway analysis. If you're interested in this work, then take a look at this issue that came out last February in Nature. It was just before COVID hit, so I forgive everybody for not seeing it because it was literally like days before all the news turned to COVID. But I guess the punchline was, you know, when you look at coding mutations, that's just in the 1% of the genome. And so this new project attempted to look in the rest of the genome. So all the different changes we see outside coding regions. And just to summarize for you, even after you've accounted for all that from all this different work from this international consortia, we still have like 10% of patient. If you look at 10% of the patients, well, you know, depends how you estimate this, 5 to 10%. You don't know what's driving those tumors. And so it's still a mystery. And for the rest of them, you might just have one that you're guessing that that's what's driving the tumor. You know, mile high overview of that project, just so you can see all the different ways we looked at it, right? You look at the structural variations that could cause tumors, mutational processes, like I just called them mutagens here. These are called mutation signatures. So different effects from UV light or toxins hitting the body and how they create mutations biased in the genome in a biased fashion. Angela Brooks led the project to look at how RNA, different types of RNAs, isoforms of splicing or alternative promoter usage and that kind of thing, how those affect tumors or how mutations affect those processes. If you're, you know, interested in line elements, retrotransposing, there's a whole paper on that, or mitochondrial mutations. What do viruses do to the genome? All this kind of stuff. And even so, I'm saying that there's 10% left over that we don't understand. And so that leads to one of the projects in the group that we still work on, and that is to try to figure out how do you relate these samples where we don't know what the driving event is. How do we actually predict what it is? Can we use machine learning to do it? And what this plot is showing you is a particular cohort, thyroid and TCGA. And some of them are driven by BRAF or BRAF pathway mutations or RAS pathway mutations. And what I'm just trying to highlight here is that you can see that many of the tumors have, is somebody talking? Many of the patients that have who look like they have cancers driven by, let's say, RAS, don't actually have a known event in RAS for that. And so the implication would be that maybe these events actually are, maybe it's the events that are hitherto not classified as RAS driving, maybe they are driving the tumors to look like RAS. And this is a slide that Angela Brooks showed at one of her talks, and I'm sort of abusing and reusing it. And the punchline is that you can create an alteration in a pathway. In this case, Angela uses the example of MET activation, but there's a lot of different ways to cause a pathway to get activated or maybe inactivated, a lot of different ways in the cells, and cancer cells find a way to do that. And so our job as informaticians then is to kind of use an integrated set of data to figure out, well, what alterations, if it's not clearly a RAS mutation, like what other alteration might be leading to this RAS activity change, whereas it's not a RAS mutation. So this relates to a project we have with the Hausler group and Elena's group called Treehouse, where we're now trying to use the RNA information as a kind of a proxy for the type of cancer, and then look at what events kind of drove that cancer. And so what Treehouse does is they take samples from a consortia of childhood cancer, the kids who had DNA sequencing and they don't know what the driving event is from the DNA, and then they get the RNA. And these are kids who have failed first-line therapy like radiation and chemo. And then the Treehouse project gets it and looks it up on our RNA maps. And you might have seen folks show these tumor map plots. That's what these colored maps are. And each dot's a tumor sample that we've ordinated, and we got it from a collection of adult cancers and childhood cancers. And then the question is, where does a new sample from the Treehouse Project map, and what can we infer about it after we've placed it in there? And this is kind of what you can think about you're doing. You're looking up, as you look at a particular patient, you can look at a neighborhood around it. And so this plot on the bottom is kind of showing you if you ordered all the other patient samples in that map according to one patient. Okay, so closer is on the left and further is on the right. What you can do is then ask, are there any events that are sort of enriched in the neighborhood, right? And so this is showing you an ALK fusion enrichment in the neighborhood of one child's cancer. This was one of our early examples that we looked at. A particular one, sorry, where that was a sarcoma, and it landed in this neuroblastoma area and had ALK fusions. And so then when you look at ALK in this patient, the green is the patient here, the expression level of that ALK looked as high as if it had a fusion in ALK that drives the expression of it, or amplified cancers and so on, and not like your typical sarcomas. And so that this was sort of a clinically actionable thing to follow up on, because there's a known pathway for that, and a drug called crizotinib that can take down the activity of that pathway. Anyway, we have multiple examples like that, and the punchline is you can use the RNA data to then get a hook about what event drove it by looking at similar tumors with the same expression profile. So I'm purposely skipping I'm going to come back to the second bullet point because the third one relates better. So if we go back to these samples where we don't know the event that drove them, we're trying to figure out what events may have given rise to the same kind of transcriptional signature. And so David Hahn, when he was in the group, created this method he calls LORE, learning unrealized events. And the kind of the cartoon we're using here is if you have a signature, so this is a gene expression signature for one particular tumor type, which he calls, he calls it the bait. So this is the signature. If you have KRAS mutations, you can build a machine learning classifier that represents this gene expression signature. Then you're looking for other cancers that don't have KRAS that have the same signature, and you're going to implicate that they also are causing the same kind of thing as a KRAS mutation. That's the idea. And David called it lore because he was totally into fishing here. That's him actually on his kayak fishing out in the bay. So what he's going to do is he's going to create a machine learning model that represents a signature for each event that we know about, like a lore mutation. And then he's going to go looking for other events that look like that, whose transcriptional patterns look the same. And he's going to call those prey events. And David's idea was you can actually iteratively expand the class of events around one that you know about. So if you know, let's say you have a particular mutation, or we'll call it an event A, and some of the and then let's say you go through and you order your samples, in this case, so the columns, by those that have this mutation A. So up here it shows you all the samples that has some event A, like a mutation in KRAS. And the pattern of data that he's calling features is supposed to show you the gene expression pattern. So you can order everybody by that event, and then what you do is you look for other examples that look like the same kind of pattern as having event A. Now if this y-axis shows you the similarity to the pattern A, you can see these are the samples that have the A mutation, but there are some samples that don't have the A mutation that also score high. They have the same transcriptional pattern, as those with A. And so, for example, if you look at some samples with event C, they also get a high score. And so David's method lore would say, well, C may be related to event A, because whenever you have event C, you also have a high score. You have the same transcriptional pattern as if you had event A. And so he goes then and he combines the samples that either have A or C together, and he repeats this process to look for an additional event. And so what he's doing is he's building this tree. So you start with event A, and he found a new event E. He finds a new event E, and then using the AE classifier, he finds a new event F, and so on. And he can keep going until he doesn't find any more events. And at the end of the day, he's actually found what we call the event net, where, depending on the order that you build these classifiers, one can find the other. And this can actually look kind of messy. So like this is what the event tree would look like. And he prunes this thing with a thing called a set cover algorithm to get rid of, like, for example, H already explains a set of samples that are already explained by other events he's found. And so we don't need H to explain any other things. And so it's redundant. So he ran this on some positive controls first. So you can, in this test, he starts with a particular mutation in IDH1. This is in low-grade gliomas. And then he's ordering by the score here of his classifier. That's what the top plot shows you. So you can find back all the things he started with. And then he also held out some IDH1 mutations, and those get high scores as well. And in addition, he's also scoring high IDH2, another family member of IDH1. He scores high the missense mutations in that, and that's a positive control because those two actually lead to the same type of cancers. He also used it to find another telomere maintenance pathway set of alterations that lead to what we think are the same kind of alterations. Which one of the things we talk about in the Nature paper that came out in February was that he implicated retinoblastoma mutations with this alternative lengthening of telomeres pathway through his lore analysis. He's run it on lots of different cohorts. I just wanted to show you that just to connect it to patient outcomes now, if you run this method again and in LGG low-grade gliomas, and you use not the HRX, but you use this inhibitor of P53 called MDM4. He also finds that high scorers, some of them have EGFR amplifications. And then if you group these patients together, the MDM4 amps or the EGFR amps, then you get a better prediction of patient outcomes in this case by, collecting those patients that together in that aggregate group. And so that might suggest that their tumors respond similarly to the therapies that they're getting. So David went ahead and he actually applied this to all the different tumor types in the pan-cancer data set, this Cancer Genome Atlas, to see what we would get. And he went through a lot of different possible things. But at the end of the day, what this is trying to show you is it's not too overwhelming, the results. So the bottom is kind of like the things that he relates, 35 events he relates to known drivers. So this is sort of, out of all this 11,000 tumors and all the different events and all this kind of things, there's really, not many more, I guess, from this coding analysis that he could pick up. So he relates these 35 different catch events. I'm going to skip over this thing just to show you the punchline that he finds some of the usual things like P53. If you start with a P53 mutation, well, you get other events in P53 like splicing changes, so that's the top one, or a P10 deletion or a P10 splicing change. I mean, I guess the cool thing is sometimes you don't know which splicing changes are driving but so he's related that at a patient specific level. He's also connected, kind of genes in the same pathway, genes in the same family, and so forth. And the overview of these events is the punchline here is that for the most part, you see one color event relating one event to another event. So like a P53 missense mutation could have a similar transcriptional signature as this alpha catenin kind of missense mutation. And there's one color, meaning he found it in one tumor type. There's some where he finds it in multiple tumor types, but for the most part, it's kind of tumor type specific, the effects that these events have and how they relate to each other. So that was kind of interesting. So the next part of the talk, we want to now kind of go back to what some of the things that Shaheen was talking about is like, how can we actually interpret these tumors? What kind of cell types are in there? Because it influences the tumor. And so we want to leverage these single cell technologies to kind of understand the bulk tumor, but deconvolute it, sort of like we were talking about at the question session. So, so we're going to leverage single cell technologies to do that. And the lab has put together some analyses to help us utilize single cell data sets so we can get a picture at some of these possibly rare cell types in the tumors. We've worked on these trajectory analysis methods that was hinted at in the previous talk. So, this is sort of my slide on trajectory analysis. It's like when the biologist collects the data, it's kind of like somebody going out into the fields and collecting all the plants at a particular time point. So you have the time of the picking of the plants, but the plants have a mixture, right? There's heterogeneity. Some plants grow faster than others. So you could even get some strawberries early on. You could get some flowers later on, but you'll get an enrichment for flowers in the beginning and so on. So the idea with trajectory analysis is use the pattern of the data, right? So in our case, transcriptional data, and in this case, the imaging, like the colors and the shapes of the plants. If you use that pattern and organize what you see by the pattern, then you can hopefully reconstitute the developmental process from a flower to a strawberry kind of thing. That's the analogy here. And so we're using the trajectory analysis uses the transcriptional data to order these things. I'm going to skip over the technology stuff. When you deal with single cell RNA data, there's a lot of issues with it. One of them is dropout. And I just wanted to plug Ding in the lab's work where he's come up with a transform to help this. So this is just my picture to show you how much missing data am I talking about. In the old days, microarray data, 3% was bad, 24% is terrible. And actually, a lot of times in the early days with single cell, we had 80% missing or 50% missing. It's always getting better with the new versions of these technologies. But anyway, there's a lot of missing data. Ding published about a year ago now this transform called biological process activity transform, where we relate the genes to the pathways that they are known to be associated in, and then we take every single cell example and replace it with a pathway representation. So we throw away the genes, the individual genes, and now we have these inferred activities through a method that Ding developed. And then the cool thing is you can actually relate them across species if you have biological pathways that are comparable, right, between, let's say, human and mouse. Ding was able to show that it increases your specificity when you compare. Sometimes when you compare RNA-seq from two different tissues, they look similar, but if you do its transform, they don't look similar, which is good. Or you can relate common tissues across species, which I'm not showing here. He did use it in his paper. You can check out his paper to align mouse and human development and show, that there's a quicker kind of development in mouse than human, and what cell stages are kind of delayed in human development, for example. So we've worked at pathway transformations of single cell data. Also, trajectory analysis, which Shaheen mentioned, where you're linking cells contiguously. So you want to actually take kind of a step back and see what the data is telling you, right? Not just do clustering analysis. Like clustering analysis wouldn't really convey the message of this particular plot. You need to kind of follow exquisitely some lower order, what we call a manifold, through the data set, a lower dimensional structure. Some of the early work, we mentioned monocle during the question session, and this is a hematopoietic development example from the monocle paper, actually. So it's finding this branch of development in blood development. So this is what trajectory analysis is trying to do: walk through the data set and find a contiguous kind of time series through it. We have a paper that will appear in Developmental Cell in the next month or so, where we looked at a developmental heart, it's a model for heart disease, where some people inherit an allele in TBX5. And so we did a single cell study in fetal development. And this is showing you another kind of trajectory analysis where you make trees out of the data. And you can then look at, well, what's different between wild type and mutant in these trees? And you can see sort of pileups of particular cell types in the mutants that aren't as dense in the wild type, in the single or the double deletion mutant in this case. And then you can ask, and I won't show this, you could look for gene expression changes that typify those populations going down one arm where they're supposed to go down another. So you can see like this kind of trident structure down here, right? There's a different number of cells getting funneled into the left sort of fork of the trident versus the right and wild type kind of thing. And you can ask yourself, well, does that underlie what's going on in heart development? Are you getting too much of this cell type and not enough of this cell type kind of thing? And what genes might be responsible for that switch? So there's lots of this data coming out, and you can look at, almost every week there's, like I'm saying here, whole organism development you can watch with single cell technologies. It's super exciting. And we're trying to develop methods to interpret that in the lab. Lucas in the lab, for example, is trying to use a transformation of the data. So you stick in single cell, you get out the same single cell. And a lot of folks have learned of this deep learning method called autoencoders. There's no deep, I'm not showing the deepness of the layering here, just to focus on the first level. Instead of just having a generic autoencoder, what Lucas is trying is you can connect these, you can create the hidden layer to represent known pathways. So, bias the genes that connect to these interpretive encodings here. And you can't do as well as a full encoder, but what we're trying to find is ones that do good enough and are still interpretable. And Lucas has some cool examples now where, if he applies it to this immune data set, he can see a lot of immune signaling are picked out. These are kind of showing you which latent variables are significant, and the latent variables mean something now. They are pathways, and so we know about them, which is kind of cool. Instead of being just black box machine learners, he can actually plot for you like a t-SNE plot, and his plots are in latent space, so they're in reduced dimensions, but you can see he has labels on them now. So he knows that this population might be interesting and weird because it's got a higher tryptophan catabolism for some reason that separates out these particular monocytes from others. And so I'm not going to show you the positive controls, but so it's cool that you can implicate in one step now by looking at these latent variables. Ding now and Bianca are collecting lots of different data sets of single cell, and this is showing you a hierarchical view. So you can cluster each data set. Each data set is kind of massive themselves, like hundreds of thousands of cells in them, and you can cluster those or run trajectory analysis and find the kind of the milestone points on the trajectories. And you can then ask yourself, well, how do you relate these across data sets or across studies? And that's what they're trying to move toward. And so what they're showing you here is when you process an integrated mouse data set and you relate these clusters to each other, what you can see is the cell types get clustered in this meta level clustering. Those are the colors on the right. And even the timing seems to be preserved in development for the mouse here after you've run their integrative kind of meta clustering approach. So we're trying to experiment with that. They have a graph clustering algorithm that relates these clusters across data sets. If you hear our group talk about single cell beacon, we were inspired by the Hausler and Melissa Klein's effort to share variants across labs. They call that the Beacon Project. We want to be able to share single cell, like cell types that we find and cell transitions that we find across data sets. And so that we were inspired by that and called it single cell beacon. Nathan in the group is trying to figure out for these trajectories, what's driving them. So you can see here's a particular trajectory again, an immune pathway example. And you can order the cells along these branches and get what we call pseudo time, right? So it's kind of like the order along a branch of a cell. And then you can look at your favorite gene in these things. Well, Nathan's idea was to figure out, is there a transcription factor driving a particular arm? And if we have known targets of that transcription factor, you could see correlations of its targets along pseudo time. And so, but those could be shifted because every target could have its own kind of nuanced expression dynamics relative to the transcription factor. And so he's applied methods like dynamic time warping or different kind of time shifted correlation metrics to try to relate those factors to each other and detect these transcriptional responses. And he calls his method Dream It, I think, right now. So you can talk to Dream It, talk to Nathan about this method. So that's another new project that we're working on. So all this single cell data then that we're cataloging and trying to organize into clusters and regulators and annotate those things, we want to then feed back into the beginning of the talk where how can we use this to learn about patient samples that are bulk. So we have these bulk tumors and they're heterogeneous. They have microenvironment cells in them and maybe different subclones of the tumor and so forth. So how can we use this single cell data to learn about those? And Verena in the group has put together a method where you take the clusters from single cell and we use methods like Shaheen mentioned, Aaron Newman and Andrew Gentles put together CyberSort, where you can give it a signature representing the cell type and you deconvolute each individual sample into the constituent cell types that contribute to that bulk mixture by running these methods like CyberSort. And so the output of it for a particular tumor will tell you, well, it thinks it has mostly the green cell type. Maybe there's a bunch of T cells in it, and the green is a T cell type that we get from the single cell clustering from SC Beacon, for example. And then what we're trying to do is take the old tumor map, which was made from a mix, from these bulk samples, which were mixtures. We want to make one that is the intrinsic tumor. We don't really know how to do that, as you heard my question from Shaheen's talk. And we also want to make a microenvironment map where we just compare tumors based on the kind of microenvironment they're in. And that's what Verena is attempting to do now. And she has some interesting examples. This is a kind of showing you breast cancer subtypes, the classic are basals and luminals, and she's relating those to other kinds of tumors based on their immune signaling pathways here that could implicate them for particular immune therapies like this. These are showing tumors that have high PD-1 signaling. How am I doing on time? So I have 4 minutes. So I'm going to just show you one quick new use to kind of lead you into. We want to be able to use all this information to predict patient outcomes. And so it kind of feeds into the machine learning, a particular type of machine learning classifier. Our bent in the lab is to use pathway information. So we're pushing on that strongly. So there's lots of pathway databases out there in the literature you can take advantage of. Some previous work in the lab 10 years ago now, people have heard us talk about Paradigm. It integrates data using pathway knowledge. There's stuff from Andrea Califano's group for transcription factor regulators, from Trajectories group to relate mutations across pathways. Vlado contributed a method that he calls AKLIMATE. We have a bioarchive and it's still under review, but it's cool because it's been shown to really outcompete a lot of different methods. So the overview for AKLIMATE is it takes a lot of different types of data, and it also will take pathway information for its machine learning classification task. Vlado developed it as part of a competition, one of these dream competitions where we're trying to predict what cells will die when you delete certain genes. Okay, that's this gene essentiality challenge. I'm going to show you that this is the results of the challenge when they run several different methods. So this is an order of the accuracy of those methods. And the accuracies here are not so great. This is Spearman correlation. So when you're trying to predict across all the cell lines who's sensitive or resistant to what knockout, what particular gene knockout, the best method's at 0.2325, and the next one is 0.2321. Anyway, they're statistically a tie, and they're all kind of miserable at doing this. But AKLIMATE, which we used to call multi-pathway learner, was one of the ones at the top. And Vlado went on to actually make it, improve it, and make it a lot better. The gist of his method, which is a really cool idea, is he uses random forests to combine lots of different types of data. Okay, so you can let the forest kind of choose decision rules that use either expression or copy number or microRNAs, or, well, not microRNAs yet, sorry, or mRNAs. It's sort of a bad plot. He couldn't use microRNAs. And then you build one of these things for each pathway. So one pathway, you build what we call a kernel. A kernel, just, you can think of it as it relates all the samples to each other. The way he converted into a kernel was he watched where do samples get classified in these random forests. And if two samples get classified similarly, and he has 3 different ways of saying what do you mean by similar, you can make a kernel. Well, these samples went to the same branch in the trees, or they got classified, they had the same labels and this kind of thing. So he has different ways of associating the samples to each other. And then he can combine the kernels using a weighting that he learns through an ensemble kind of approach. And so he does well at predicting knockdowns. He's, competes with the best methods out there. And then I guess the cool thing is this is a typical plot from AKLIMATE. If you order the cells by their essentiality to a knockout, in this case, we've knocked out FOXA1, which is a kind of a chromatin activator involved in AR. Actually, what you can see is, this is a busy plot. You see that there are features he's picking up from multiple different tumor types, from different data types, that predict the essentiality. So if you have like high expression in a particular cell line of FOXA1, that's this one, if you could look at this gray box, then you're going to be more sensitive to knockdown. Your essentiality is lower. Your essentiality is more sensitive to the FOXA1 knockdown. Sorry, my alarm's going off. I'm saying it at that time now. But you can read this out from Vlado's method. So if you use different types of data and things in the FOXA pathway, including AR, it's a pioneering factor for androgen receptor. And we can look at the pathways that that AKLIMATE locked in on. It's being used now in a current project for the Cancer Genome Atlas, where we've categorized all the patient samples into groups, and AKLIMATE's being run now to try to find a minimal predictor. So in this situation, you're trying to classify all the samples into each of their subtypes in each of the cancers, and you're only allowed to use a certain number of features. That's what this x-axis is showing you. If you use all the features, you get the best accuracy you can, right? But we've been given a rule because we want to make a clinical test that we can only use, say, 50 features. And so the best classifier gets 88% accuracy or 89% accuracy when you use particular 50 features. So AKLIMATE's being used for this. I'll skip this other example. And just to plug that, AKLIMATE's the first plot here, and we're looking at each of these are different tumor types and predicting the subtypes within the tumor types. And it's showing you the data that's used in many different algorithms. And the long story short, AKLIMATE's not as good as other methods when they get to use microRNAs. And so we're looking at how do we actually use microRNAs. So there's a project if people are interested in helping us better AKLIMATE to use microRNAs. So I guess I'll stop there. The summary of the talk was there's lots of tumors we don't know what's going on still. I mentioned this 10% number. That has no known driver, even for the ones where we were predicting a driver. Maybe we don't really have a handle for it. mRNA we use as sort of a signature of the pathways or the cell types involved in the tumor process. And I showed you lore that can relate events to each other using the mRNA signatures, or AKLIMATE's used for outcome prediction. So we have machine learners to do that, and we're trying to leverage single cell data to deconvolute tumors, and that's primarily from mRNA. Ding's pathway inference helps us kind of take advantage of that stuff, and AKLIMATE uses pathway information. We're trying to build toward, in the next push in these consortia is to come up with methods that can predict patient outcomes now. We're still miserable at doing that. Two tumors look like they're the same to us, and they have very different outcomes depending on treatment. So I think I'll stop there. Maybe I don't know if I have the picture of the lab again. There we go. And